Okay, moving on to the next step. This is how the siRNA pathway would occur. So the double-stranded RNA, when it enters the cell, there's a ribonuclease enzyme. This is an endoribonuclease enzyme, just like the endonuclease enzyme, restriction enzyme. But this would always cut a double-stranded RNA, not DNA. So that's the difference between the dicer and, let's say, EcoR1. It will cut this double-stranded DNA into shorter pieces. It will still be a duplex form. And when this relatively longer double-stranded RNA molecule is cut into shorter pieces, it will be recognized by a complex which is known as RISC complex. And what will RISC complex do? RISC complex is actually a combination of a number of protein, and what it will do, it will degrade one of the strands. This is having two strands, the blue one and the red one. When the RISC complex recognizes these short double-stranded pieces of RNA molecules, it will degrade one of the strands. Which one of the strands will be degraded? There is a selection process, and whatever RNA strand is thermodynamically less stable, that will be degraded, and the other will be not degraded. Now, designing siRNA, it is very important that the strand which you want to, like in this case, is the red strand, that it has base complementarity towards this messenger RNA. So we have to design it in such a way that this red strand is thermodynamically more stable than the blue one. Only then this RISC complex would be degrading the blue one and leaving behind the red one, which will have base complementarity towards a specific messenger RNA. Once this single-stranded messenger RNA along with some proteins, when they bind to the messenger RNA, that messenger RNA would be chopped in a number of places and the translation would be inhibited and the gene would be silenced. The two important proteins that we discussed here is dicer and RISC. So this is the structure of 3D structure of dicer, and this is the 3D structure of argonaut. Argonaut is one of the most important protein of RISC complex. In RISC complex, there are argonaut. There's two domains, the PAS domains and the PV domains. And as I said, dicer is an RNA, dual RNA, 3 domains it has, and it has a dual double-stranded RNA binding motifs. These are just some structures you could see. Mostly they are alpha helices in the structures with some occasional beta sheets. We do not need to go into the detail, but if we compare the two structures of dicer and argonaut, we could see that argonaut has more beta sheets. These, like you can see, the beta pleated sheets. Now the same process will happen if we have a double-stranded RNA. This is the small interference RNA or the short hairpin RNA. In both cases, in short hairpin RNA, we will design it in such a way that there is base complementarity and a hairpin loop is formed. The dicer will recognize the double-stranded RNA molecules and it will chop it into smaller pieces. Then what will happen? The RISC complex, which is argonaut with some more proteins, it will bind to the smaller pieces. Whatever RNA strand is thermodynamically less stable, it will degrade. And then with the single-stranded messenger RNA bound to the RISC complex, it will find its relative messenger RNA molecules where it has complete 100% base complementarity, and the siRNA and the messenger RNA complex will form. Once this forms, the RISC complex along with other accessory proteins will chop the messenger RNA And when the messenger RNA is chopped into pieces, it cannot be translated, and the protein for the protein synthesis for which the information is contained in this particular messenger RNA would not be made, and we will have our gene silenced. Huntington disease is a neurodegenerative disease, and could have mental capacities loss, movement loss, and also one of the symptoms is temperament. You would lose your temperament so easily. When we see the normal gene, it has 10 to 26 CAG repeats. This is the gene. But when there is mutation, you would see that this 10 to 26, it will jump 35 or more, which means that when this mutated gene is translated, you see the green parts are the normal ones, but now there are more CNG repeats coming into the gene, in between the gene and the expanded polyglutamine protein. It will be a polyQ tract. It will be not as the native protein is, and this is the reason why it is caused, or how it is caused. How do we fight diseases, for example, Huntington disease, through RNAi or to siRNA molecules? Now we see that we have a normal gene pool, then there could be a genetic mutations. When there's a genetic mutation, the gene is mutated, we call it a mutated or mutant gene. It will make a messenger RNA, and we will have an altered protein. In this case, this protein is altered because this light blue part is the extension or the extra part of polypeptides that should not be here. Only this green should have been bound to this green, and it will be normal, but it's not normal. So we have an altered protein, and then the pathogenesis. Pathogenesis means that the disease will cause its pathogenicity, it will cause the disease, this protein, and eventually we will have symptoms, the progressions of a disease or a disorder. This is the normal way of from normal gene pool through genetic mutations, through pathogenesis and symptoms. Now, where can we use RNAi? Right here at this step. Synthetic siRNA or shRNA, we will design, and we will inherit, we will target the messenger RNA, or you can also use miRNA, and they will knock the muted gene. So for this protein, there must be a messenger RNA molecule. And what we can do, we can design siRNA or shRNA that will go and bind to some part of this messenger RNA, or more than one part is also possible. And eventually this messenger RNA would be degraded. So that's the Huntington gene. That's the messenger RNA. And with gene silencing, the protein would not be made because the messenger RNA will be chopped before it's being translated. Now, how do you do this? You have siRNA, or you can also call them double-stranded RNA molecules. Now, the thing here is very important. This is why I was not following the order as mentioned in the course catalog for your course, but rather I was trying to make a logical order to follow for the lectures. Now, there's a plasmid, right? And as this plasmid, we have cloned a gene that will have that will be encoding the RNA molecule, the double-stranded siRNA molecules. And you see that this plasmid will go inside the cell. Now, what is a plasmid? We know. How can we clone it? We know. How can we enter the plasmid? We know. How can we use plasmid for higher eukaryotes? We know. All these things we know. And this is like the gene therapy, all the techniques we had in gene therapy, in genetic engineering and transgenic animals. And this is kind of a gene therapy. shRNA is a part of gene therapy for some cells which are cancerous. You can design siRNA for those particular genes, and their messenger RNA would never be translated, and the cancer would never progress. Now, there are two methods to do it. Either you do it through gene therapy by inserting the gene in a viral vector and then integrating it, that part of the DNA, into the genome. So the messenger RNA, which are repeatedly produced by the body, the same will be the case for this integrated piece of siRNA. They will be repeatedly produced by the body, and there will be gene silencing going on forever. So shRNA or siRNA, it does not make any difference, will be continuously produced, and the enzymatic cleavage of the hairpin or the double-stranded, just like in this case, you see either it's a shRNA or it's a dsRNA or shRNA, it's the same case, nothing will change. And the siRNA, the enzymatic risk complex, will, when the dicer has its effect, making it into smaller parts, it will always be recognized by the risk complex, and the risk complex will degrade one of the strands, and the other strand would always be remain associated with, and eventually it will go and attach to the messenger RNA, where it has complete base complementarity, where the attachment happens exactly there. The messenger RNA will be chopped into pieces, and because it will be chopped into pieces, the protein would never be made, and if the protein is never made, the disease symptoms will be controlled or neutralized. So as long as there's enough supply of these RNA molecules, this gene would be silenced. This is why we say it's good, a good and a better approach is to genetically transfer the genes for the synthesis of these siRNA or shRNA into the genome, so that as long as the organisms live, it is continuously being produced. Otherwise, you can also design this siRNA or shRNA in lab, and then you can inject them into the cell. But in this case, you would have to work like a dose dependent. After every 3 or 4 or 5 hours, you have to inject a certain amount of siRNA molecules, because they are not made naturally by the cell. But in this case, they will be made just like all other RNA molecules by the cell themselves. And we just discussed that you can design and produce and purify these siRNA or shRNA outside the cell in a lab, and then inject it into the cell for gene silencing. How can you do this? So this is a liposome. You can do it in a liposome, or you could do it with other nanoparticles that are loaded with siRNA molecules. I have recently designed a similar kind of project for some studies at Max Planck Institute and Potsdam universities, along with some collaboration with the Freie Universität Berlin. If these studies, if that proposal is funded, I would go for research for 2 months, no, for 2 years in Potsdam University Department of Biology and Biochemistry. But if it's funded, the results will come in October, and I have been using more or less the same strategy. The rest is the same process, you can see. So in the nucleus, you have the messenger RNA, and the siRNA are encoded in these lipophilic nanoparticles. And I know these are not nanoparticles, but it's a nanoparticle here, nanoscale systems, and eventually it will cause the cleavage of the messenger RNA, and there will be no protein made. Okay, as you all know that I have given you a small, not a very small, but another assignment of therapeutic proteins in yeast and insects, and the third part was in E. coli. I did not mention it because discussing E. coli in this lecture is killing 2 birds with 1 stone. It will serve 2 purposes. You would have that topic covered, as well as we would see how can we link it to siRNA or RNAi technology. So this is the structure of a complete molecule of insulin. We know it has a total number of 51 amino acids, and the 51 amino acids are actually divided into 2 chains, the alpha, not the alpha, but the chain A and the chain B. And these dots that you could see, these are disulfide bridges between the cysteine amino acid molecules between the two chains. So the two chains are held together by disulfide bridges. When the insulin is synthesized, it is synthesized as a pre-proinsulin, and you could see when the pre-proinsulin is synthesized, it is a signal peptide, it is a B chain, C chain, and an A chain. Once this is made, there is a cut. So the first process is the signal peptide is cut, because the purpose of signal peptide is to take it to transport it to particular location in the cytoplasm. Then the removal of signal peptide is followed by another cut here for the C chain, and the C chain, this C chain is cut, and eventually there will be another cut, because you will have to cut it here and you will have to cut it here, so that the two insulin chain A and chain B could be bound together. And the removal of the C chain, so you have the proinsulin. From the proinsulin you have the insulin with the B chain with 30 amino acids and A chain with 31 amino acids, and held together by disulfide bridges. Okay, how do you synthesize this recombinant insulin in E. coli? So you take A gene and clone it in promoter, and then you have the B gene and you clone it in not promoter. So you take the A gene and you clone it in, I suppose, plasmid A, then you take B gene and you clone it in plasmid B. Since the A chain is 21 amino acid and the B gene has 30 amino acids, that's why it has to be larger in size. You transfect or transform E. coli with it, and you culture them, and after culturing them, you just retrieve the protein through this MET. This is a signal peptide. This is how you can harvest and clean or purify the protein. And after this purification, you, in this case, it's a MET signal peptide, and with the cyanogen bromide treatment, it can be cut. So when it's cut, you have the cleaved fusion proteins, the B chain and the A chain. Now what you do, you purify the A chain and B chain, and you mix them in equal proportion. So if you have, let's suppose, 5 molar of A chain, you will have 5 moles of B chain, and you mix them in vials and mix them under ample amount of oxygen. So when there will be ample amount of oxygen, these chains will be together because of oxidation, and they will give rise to these disulfide bridges. And eventually you can have the insulin, which can be injected into the patients of diabetes. Now, when we talk about diabetes are of 2 types, type 1 DM and type 2 DM. In type 1 DM, there is no insulin made whatsoever. So there, the only treatment is insulin. But in type 2, the insulin is there. There are 2 very important characters in the type 2 diabetes mellitus, GLP-1 and DPP-4. It's a glucagon-like peptide 1, it's the name. What it does when the blood glucose, when the amounts or the levels of glucose rises in the blood, it induces it induces the synthesis of insulin, so that the insulin could be released into the blood, and the blood sugar concentrations could be taken down, could be decreased, because when the blood sugar concentrations are high, it's not good for the body. There is another molecule which is known as dipeptidyl peptidase. It is actually an anti-GLP-1. So its action is that it will degrade GLP-1. In case of type 2 diabetes mellitus, using RNAi technology, is there any possibility that we can have a treatment of type 2 diabetes mellitus? You see, incretins, GLP-1, and then there's another protein, GIP1. What they do, they inhibit the glucagon release. So for the moment, we can just ignore this. They stimulate insulin release so that the blood pressure is lowered in the glucose concentrations are lower down in the blood. But these enzymes, they are like have a function which is anti-GLP1. So it's a continuous chain. When you want to have a treatment for T2DM, what could you do? Think about it, and we will discuss it in the class tomorrow in the interactive session. It will just take 5 minutes. Just think out of the box. If nobody could answer this, I will tell you the answer. So don't worry about it. It's not some kind of assignment. Don't hit me back with like, we are working 12 hours of job, everyone is not in the quarantine, blah, 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 blah. If you cannot do it, just think about it. If you cannot think how can you manage it, you can just come to the lecture tomorrow and we will have it. So that was all for today's lecture, and I will see you tomorrow at 1:30, and hopefully by the end of next week, the next 3 lectures, in the next 4 lectures, we will complete the course, and it will be a bye-bye to the biotechnology course lectures. Thank you. See you guys tomorrow. Allah Hafiz.